I had discussed with you the gene arrangement for the lambda light chain, the kappa light chain, and the heavy chain gene for the immunoglobulins. And this type of gene arrangement in the B lymphocyte, that is pre-B cell, okay, is called as the germline gene arrangement. It is there as a germline, GERM, germline gene arrangement. From these several genes that are meant, that is exons for the variable region and the exons of constant region that are meant, when they recombine, they will produce a functional receptor on the B lymphocyte, and that receptor may be productive or non-productive. The productive one will be with the low affinity or high affinity, and non-productive will be without any affinity. So this recombination between variable and constant region genes, we will see today. The germline gene arrangement shows recombination to produce the receptors on the B lymphocyte. First, I will take up the kappa light chain gene rearrangement and the recombination. So, the variable region genes that are present in the 5' end in clusters. Now, how to recombine? By joining with the BJ and then with the C, and that recombination is called as VJC joining. First, the recombination takes place between any one of the variable region gene with the J segment, that is called as VJ joining, and then the VJ will join with the C segment. And the joining of the segments takes place by formation of a loop. Now this V will form a loop like this and join with the J. Okay, a loop will be formed, a hairpin loop like structure will be formed, and the in-between genes are going to be cut off or deleted, lost. Every time when the recombination takes place in this segment, it is in the RNA, that is the recombination that is taking place in the product that is produced. Therefore, in the splicing, when it happens, the recombination will take place in such a way that some of the segments in between which were coded will be lost. Okay, it is not the gene that is going to be lost. It is the processing of the RNA. Okay, the recombination is in terms of joining of a functional segment, that is exon, with another exon. So we'll take this example here. In the diagram, it is very clear. The gene that is chosen is here. They have shown the recombination of VK23, that is 23rd gene of the variable region exon, with a leader sequence of its own. Okay, is recombining with the 4th segment of the joining segment. Okay, so that in between segments are now lost. So the joining takes place in such a way that VK23 has joined the 4th, the joining segment. Remaining J1, J2 and J3 anyway was not functional or lost. There is one additional junctional segment that is existing between the constant region segment and now VJ, which has rejoined. Now it has to undergo once again another recombination to produce a primary transcript, and that primary transcript will be in such a way that VJ will show one more loop with the constant region, and the in-between J segment will be lost. Finally, it will have constituted a segment of VJC. So I had said that for the light chain, it is always VJC, and for the heavy chain, it is VDJC. Okay. variable region, diversity region, joining region, and constant region for the heavy chain. Whereas for the light chain, whether it is lambda or kappa, it is VJC recombination. The final mRNA that is going to be produced. This process of cutting and joining is called as RNA splicing. All right, you have done that in molecular biology using spliceosomes. All right, the cutting and joining is brought about by the spliceosomes. So final product that is going to be produced, which is going to be a transcript, and that transcript, that is the messenger RNA, will have the final combination of the exons that are ready to be transcribed and translated into a polypeptide. Okay, translated into a polypeptide. Now this transcript will translate a kappa chain, and that kappa chain will be the kappa light chain for that particular receptor that is going to be used by the B lymphocyte. So this independent event, that is light chain recombination event, is taking place independently on chromosome number 2 as well as chromosome number 22. Chromosome number 22 is for the lambda chain, chromosome number 2 is for the kappa chain. Each time when the B lymphocyte is proliferating, undergoing mitosis, this gene rearrangement is going on to put on the receptor on the surface of the B lymphocyte. Okay, so one product that I'm showing you here is the kappa light chain product which has been produced. Now let us see how the recombination is taking place simultaneously at the same time in the same B lymphocyte in the heavy chain gene, that is on chromosome number 14. In the chromosome number 14, first joining of the segment will be DJ joining. D joins the J segment. So any one of the D segment, here I have mentioned once again, it is the mouse genome that is shown. In the human, the diversity segments will be about 30 and the joining segments will be about 4 to 6. So any of the 4 to 6 joining segments can be chosen of the 30 diversity segments, any one of them. So let us presume that DH7 means 7th segment of the diversity is choosing the 3rd segment of the joining segment. So there will be a loop formation between these two, and in between segments will be lost and DJ joining first takes place. After the DJ joining, it will facilitate now picking up of any one of the variable region genes. Now here in the diagram, they have shown the VH21. Okay, and that is going to recombine with the DJ segment. With the result, the in-between segment that was there is going to be lost. The splicing takes place between the variable region, one of the segment, with DJ segment. Now here VDJ joining has taken place. Similar to that we had observed, there is one additional J segment that needs to be lost. It will be lost when this VDJ segment first recombines with C mu. Okay, because always the first receptor that is going to be put up on the B lymphocyte will be the IgM receptor, membrane bound monomer IgM receptor. The second immediate receptor will be IgD receptor. So how it happens, let us see. Now these two genes, constant region genes of the mu and delta, have to show recombination with the VDJ. That is what is shown here. VDJ is going to show two different recombinations, VDJ with C mu, VDJ with C delta. So that recombination can be represented like this. You have now VDJ C mu, one heavy chain gene combination, another heavy chain will be VDJ C delta. So you have now two heavy chain gene products. These two heavy chain gene products are the first one to be expressed always. First receptor on the B lymphocyte will be always heavy chain polypeptide going to be expressed. After that, the light chain is going to combine there to form the light chain with the heavy chain combination. So let us say this kappa chain, which has been expressed in the B lymphocyte, that is pre-B cell, and this heavy chain has been expressed choosing the 21st variable region gene with the CMU, and that 21st variable gene has combined with the constant region of the heavy chain of the mu, and 21st variable region gene has combined with the constant region of the delta. VDJ combination is there, where 13 amino acid sequences of the D segment, 13 amino acid sequences of the J segment have contributed to the variable region segment. Okay, now these will this transcript will translate into form the peptide that will be expressed first on the B lymphocyte, which will now form a bond with the kappa chain or the lambda chain that has been expressed separately by the two different chromosomes. Now, once these for a proper receptor on the B lymphocyte, that B lymphocyte is ready for now receiving the antigen. The antigen has to bind to the FAB site of uh, the receptor. So when the final product, the product protein is produced, final product, the leader sequence is lost. Leader sequence is meant only to direct the recombination in the 5' to 3' direction. Once all those recombinations, splicing, everything has been taken place, next the leader sequence function is over. It is to be lost in the final product of the protein. In the final product of the protein, it is removed. And that is what is called as post-translational modification. In the nascent polypeptide, first peptide when it is going to be produced, the leader sequence is there. It is there. But when the final product of the polypeptide is dispatched, the leader sequence is going to be removed. And that is post-translational modification and processing. Here now, the recombination that is taking place in an orderly fashion, that is the 3' phase of the variable region shows recombination in such a way that 3' phase of the variable region joins the 5' region of the DH. Same way, 3' region of the D joins the 5' of the JH. Same way, 3' region of the JH joins the 5' of the C mu. It is in the same order, 5' to 3', 5' to 3'. If it has to take place in an orderly manner, there should be something there directing this orderliness. And when it is observed, it was found that there are specific nitrogenous base sequences in the 3' end of the variable region. Similarly, 5' of the DH and 3' of the DH, and 5' of the JH and 3' of the JH. And those sequences which are present there are called as recombination signal sequences. Recombination signal sequences are called as RSS. Recombination signal sequences are called as RSS. So these RSS signal sequences are specific nitrogenous base sequences that are present. They are classified into two different types. One is two-turn RSS, one-turn RSS. What is meant by two-turn and one-turn RSS? You know, in the DNA molecule, one turn refers to 10 nitrogenous bases. Two turn refers to 10 plus 10, 20 nitrogenous bases. Okay, two turns of the helix, one turn of the helix. Based on that, these recombination signal sequences are classified. The specific nitrogenous base sequences that exist on the 3' end of the variable region, then 5' as well as 3' of the diversity region, or 3' and 5' of the junction. They could be following this rule, thumb rule, of two-turn sequence or one-turn sequence. One turn means there are hardly about 10 to 12 nitrogenous bases. Just it need not be exactly 10. One turn refers to 10 to 12 nitrogenous bases. That is base pairs. Similarly, two turn is supposed to be 20 nitrogenous bases. It could be 20, 23 nitrogenous bases. Okay, and they are involved in that recombination. Now, how these sequences exist, let us see. Two turn sequences are shown by blue symbol. One turn sequence is shown by red symbol. Here, let us take the lambda chain. In the lambda chain, the 3' end of the variable region lambda chain has two turn sequence, and the 5' end of the J chain, J is supposed to be having one turn sequence. So it need not be the same rule for the another. Here you see it is opposite. In the case of Kappa chain, the 3' end of the variable region has one turn sequence, and J has two turn sequence. Now the beauty of this is it is the splicing or the recombination signal sequence is such that it is always the fusion or recombination between two turn to one turn or one turn to two turn. It is never between one turn to one turn or two turn to two turn. I hope you understood. When the recombination signal sequence occurs, it will be the recombination between two turn to one turn or one turn to two turn, and never between one turn to one turn or two turn to two turn. That means there is a regulated signal and it is an orderly signal in order to facilitate the splicing and recombination. Here in this case of the heavy chain, just see the variable region of the heavy chain at the 3' end has two turn sequence, whereas the diversity region on both the sides it has one turn sequence. On both the sides, that is 5' end as well as 3' end, it has one turn sequence. But the recombination here between variable region and D, that is diversity, two turn to one turn. In the case of J of the heavy chain, on the 5' side, it has got two turn. So recombination is between DJ, that is, will be one turn to two turn. Okay, so when this splicing and recombination that takes place, which is regulated by these nitrogenous base sequences that are present, and predominantly you see here it is AT rich region. It is AT rich. The region is more of AT. Because of AT rich region, because of the two hydrogen bonding pattern, the region becomes easily vulnerable for break and join. Break and join. Okay, so that is the reason it is predominantly AT rich region. And you know that the promoter regions of the genes also have AT rich region. Is it because the hydrogen bonds between the AT has to be broken to open the promoter for making the 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 proteins that are responsible for the transcription to be bound there. Okay, whatever may be the case, now I hope you understood the two turn RSS means recombination signal sequence and one turn RSS that regulates the fusion or splicing of the segments, exons, in the order of 5' to 3' order. Okay, it will never be reversed. To maintain that order, these signal sequences are there. Now let us see how when the recombination takes place, sometimes it will turn out to be productive and sometimes it turns out to be non-productive. Let us follow this sequence very carefully. Recombination signal sequence, they have shown some nitrogenous base sequences here. and this is in the variable region of the Kappa chain. Take the nitrogenous bases in triplets. Triplets represents the codon. Okay, in triplets, and this is the J segment. Okay, VJC recombination we are talking about. When V is going to join the J, and the recombination when it takes place, it can pick up the recombination in such a way that it can be end-to-end recombination or in-between recombination. Now let us follow these numbers. Let us follow these numbers. The first one, you take the first one. Here they have shown the combination and we have to match that with the combination here. GAG, that is GAG, represents glutamic acid. GAT represents aspartic acid. Now then the recombination is, that is GC. GC is here. GC is here. And that segment, GC segment in the first recombination has gone and joined here. Okay, so it becomes GCG. So the GCT segment, in fact, the recombination has taken place in a way GCG here. So GCG represents alanine. Next immediate will be ACT, it is there ACT, and ACG, it is there ACG. So this is one productive combination codes for these amino acids. That is first. This is one recombination. Now second recombination, suppose second splicing took place. Take the second one. Once again, GAG represents GAG, GAT represents GAT. Now here GGG. Okay, the second one G, G, G. Okay, GGG, which this amino acid and this amino acid are different. Then it follows ACT, ACG. These two amino acids are same and these are different. Now do you realize when the same B lymphocyte shows this type of recombination, suppose if this is involved in the constant region domain, VK variable region domain and constant region domain. Variable region domain, it has differed by these amino acids. It has differed by these amino acids. There is in fact one amino acid difference. That itself makes this FAB site of the variable region of the kappa different for different antigenic determinants. Now let us take the third one. Third recombination is here. Okay, third recombination here. GAG is there. GAT is there. Okay, then the recombination has taken place from here. That is TCG. So it has combined from here to here. This is deleted. So TCG. TCG comes from here. It is here. Then following that will be the next sequence. After that will be the ACT. Okay. It is not TCG, TGG. Then the ACT and AG. See now what way it has differed. Three different products are produced. Three different light chains are there. Three different productive light chains are there. This is the possibility in the recombination. Let us now follow fourth and fifth in the next slide. Fourth and fifth in the next slide. See here. Fourth one. Once again, this GAG, GAT. Okay. The GC, that is the fourth one now. Fourth one. GC is going and joining here. That is GCG. Then comes GAC. This will be used as a frame. So GCG. Then GAC. That will be used as a frame. GAC. Then comes tag. That is the sequence. When it comes, this will be the termination codon. Compare this sequence with this sequence now here. These are productive. This is unproductive. The termination of protein chain takes place much earlier because of the frame shift in the recombination that took place. And the fourth possibility that I am showing you, there is no change in the nitrogenous base sequences. It was the same sequence that was available. It is only that the recombination pattern or splicing pattern changed. When the splicing pattern changed, some amino acids remain the same. Okay, some amino acids remain the same. Here, glutamic acid, aspartic acid, alanine is the same. Okay, but the third amino acid is changed. Then the fourth is not going to be formed because it is a termination codon. And this protein chain is going to be aborted much before it is going to be produced. Similarly, take the fifth one. In the fifth one also, it is non-productive. GAG is there, GTG. Now that G is going to recombine here, GTG. New amino acid has been put. Then the next sequence, GAC, which is already there. Then comes the tag, which is stop codon. So this termination is much earlier than the previous one. Fourth and fifth, they are differing. Once again, these two are aborted sequences, means they are aborted protein productions. This protein is not going to be complete. This is not going to be productive. Okay, so that is how the recombination has taken place in the same sequence of the nitrogenous base in different format. When the recombination takes place, because of the recombination signal sequence, it will result in some productive peptides, which are here. 1, 2, 3 are productive peptides with difference in amino acids. Okay, difference in amino acids. And 4 and 5 are non-productive peptides, which are useless to form the light chain of the kappa chain. So same thing could be repeated, happen in the lambda or in the heavy chain gene, and that will result in the non-productive proteins, which will not be able to construct the receptor on the membrane of the B lymphocyte. And if this happens repeatedly in a B lymphocyte, where more of non-productive peptides are produced, that B lymphocyte undergoes apoptosis. Okay, which is a very, very rare chance. There is always a chance to have a productive peptide. And when that productive peptide produced, among these three different productive peptides, one of them may be successful in showing high affinity interaction. Okay, and one of them may be successful in showing low affinity interaction, and another may fail to show any interaction because of that particular amino acid constitution in the FAB region. And if it shows no affinity, then that particular B lymphocyte which has chosen that kappa chain has to undergo apoptosis. So that is how the protein products of the kappa chain and the heavy chain determine the survival of the B lymphocyte. And that is how the recombination or splicing takes place in the genetic segments of the variable region with the junctional segment, J segment, and that is the predeterminer of the production of a productive peptide or non-productive peptide. Okay, now we will come to the terminology called as allelic exclusion. I had mentioned this and I had told you that keep this in mind when we spoke about the B lymphocyte that has been now chosen to produce the antibody, because it has already established the first contact with the antigen with its receptor on its membrane, and that antigen contact might have taken place with low affinity. Now it has to now choose the gene that had shown the low affinity interaction to form the antibody production. Okay, and that antibody production which is going to take place will have high affinity receptor going to be put up with the permutation and combination. So for any given individual, in his somatic cells, he will have inherited one complement of gene from the mother, one complement of gene from the father. Okay, now here the color combination is shown for your understanding purpose. Please note, in the cell, the genes are not colored and they don't exist in the colored combination. They are chosen randomly, and when the choosing takes place randomly, there could be productive, non-productive gene combination. Here, they have shown kappa gene, lambda gene, and heavy chain gene in pairs. That is maternal and paternal chromosomes. That is heavy chain gene on 14th chromosome, having maternal and paternal, lambda chain gene on 22nd chromosome, paternal and maternal, kappa chain gene on 2nd chromosome, maternal and paternal. Different color combinations they have shown. Now, this is a pre-B cell or pro-B cell, let us say. It is not a pro-B cell. It has not yet put the receptor on its membrane. And this is the germline gene sequence or germline gene organization. Now, that pro-B cell undergoes proliferation to form the pre-B cell, and it will put up the first receptor on its surface, that MIGM receptor, let us say. Okay, as it is chosen to form the pre-B cell, it is going to put up the first receptor, that is MIGM receptor. In this MIGM receptor that is chosen when this pro-B cell has undergone, where we had dealt with the B cell maturation, I had explained to you this process, it has put up the first receptor. Now let us see here what is the combination of the receptor. In this, they have shown the color combination. Both the chains, that is heavy chain and the light chain, are derived from the maternal gene complex. Heavy chain is also derived from the maternal, and light chain, kappa chain, is also derived from the maternal. And what happens to the other genes in this? They are silenced. They are silenced. Those genes are going to be silenced in this B lymphocyte, which has been now chosen to produce this receptor. Okay. Now take this pro-B cell. In this pro-B cell, the receptor expressed is quite different from the one which is expressed on your left hand side. In this, what has happened? The maternal gene that is chosen for the heavy chain and the light chain, lambda chain, paternal gene is chosen. Now the receptor that is present here, MIGM receptor here, is different from the MIGM receptor here. Now I hope you understood. In the process of mitotic proliferation and two B lymphocytes are produced as a product of one cell, both those B lymphocytes need not have the same type of receptor. They will differ from one another. And this B lymphocyte will now further proliferate to put in the same type of receptor. This B lymphocyte will proliferate to put in the same receptor. Now, receptor here is different. Maternal heavy chain, paternal lambda chain. Here, maternal heavy chain, maternal kappa chain. Now, these B lymphocytes have to show interaction with the antigen. In that case, what has happened now? Among the pair of genes, only one gene is chosen. One lambda gene is chosen here, and one heavy chain gene from the chromosome number 14 is chosen. So the heavy chain gene of the chromosome number 14, that is the paternal gene, is silenced. Here, the paternal lambda gene is chosen, maternal gene is silenced in this B lymphocyte. Here, once again, the kappa chain gene, that is both paternal, maternal, are silenced. In this B lymphocyte, the heavy chain gene, maternal is used, paternal is silenced. In the light chain gene, lambda gene is not chosen, so it is silenced. In the kappa chain, maternal kappa chain gene is chosen, paternal is not chosen, so it is silenced. So when among the pairs of genes, whichever the gene is silenced or not chosen, that is called as allelic exclusion. Among the pair of alleles for the kappa chain or for the lambda chain or the heavy chain, only one allele is going to be chosen to be put up for the receptor and to be preparing for the antibody. Other pair of the gene, that is other allele, will be silenced and it will be excluded in that particular population of the B lymphocyte, and it will remain silenced until that B lymphocyte commits itself for the particular antigen. If it doesn't commit for the antigen, that B lymphocyte has to undergo apoptosis. Another new batch of B lymphocytes are to be produced from the pro-B cell. So remember, the gene that is not chosen is called as allelic exclusion. And that is what happens. Both the pairs, that is of the genes of maternal and paternal, cannot express to produce a receptor on the B lymphocyte. Any one of the pair will be chosen to produce the receptor. Okay, now coming to the diversity. Just refer to this table. You will come to know that just in the case of human beings, in the human antibodies, the heavy chain, Kappa chain, light chain, the number of functional segments, exons that have been given here. 51 heavy chain exons of the variable region gene and 40 exons of the Kappa and 30 exons of the variable of the Lambda. Diversity, I had said about 30 genes are there, 30 segments are there, of which 27 they have shown here, and there is no diversity segment in the Kappa and Lambda chain. Junctional, 6 I had said here, 5 here, and 4. Now you take any one of the permutation and combination and put them together. Now 51, 27, 6, this is the product. 40 into 5, this is the product. 30 into 4, 120 product. Now you take this and multiply. What is the possibility of antibody production? It is shown here, 2.64 into 10 to the power of 6. But it is not possible to get those many antibodies because sometimes the recombination is going to be a failure, which I have already told you. At any given instance, the antibody produced will be less than this number because the recombinations are always not successful. Now coming to the last mechanism, that is class switching mechanism. First, we mentioned that in this slide I had shown you how the mu and delta gene is going to be chosen for the production of the mu and delta receptor, that is IgD and IgM receptor on the B lymphocyte. Start with the B lymphocyte will have these two, and it will start with the production of IgM antibody, which will last for 5 days. Then it has to be taken over by the IgG. And how that class switching takes place? Of the IgG, it can choose any one of the 4 segments of the IgG molecule. Okay, now S refers to signal sequence, recombination signal sequence, S. Okay, and those recombination signal sequence are the one which are facilitating the formation of the loop, that is for the exclusion of the genes in between. Now you see here, VDJ has already been constructed, and now that has to combine with the constant region. Initially that VDJ had combined with mu and delta. That is over. Now next population of those B lymphocytes that were chosen for the antibody production, they have to start producing the IgG antibody. For that reason, it has to show once again recombination of that same chosen allelic gene, whichever the receptor that was chosen here. By excluding other alleles, it will now recombine in such a way that that gamma, which is going to form a recombination, that will be VDJ gamma 1 recombination, and other segments that are there are going to be now in the form of a loop and deleted. This is deleted segment where C mu, C delta and other segments are deleted, and the recombination has resulted in VDJ combining with the C alpha 1. Now let us say it is going to go for second recombination because that B lymphocyte has decided to produce Ig epsilon, that is IgE. These segments have to be deleted, and that will happen like that here. And the recombination once again VDJ with signal sequence of the epsilon will take place, and the segments in between will be deleted and resulting in C epsilon. Okay, so that will result in the production of now IgE antibody. So this is how the class switching takes place in the antibody production. Whichever the receptor that was chosen with low or high affinity, it will finally has to decide to produce the antibody IgG, and that IgG will be showing this class switching mechanism from mu and delta with the same VDJ combination which was used for the production of IgM and IgD, which had shown here. VDJ same will be used for recombination to produce Ig gamma or Ig epsilon or IgA, which is called as class switching mechanism. Okay, so that is as far as the antibody is concerned. And in the next class, I will make the comment about the somatic recombination that I will just mention in the beginning, and then I'll proceed with the another lecture. Thank you very much.